We're looking for data down. In the meantime, I'll show you my plants because I haven't practiced with them, so I'm not sure how well I'll integrate them. These are oxalis that we have out in a greenhouse at Washington Street, Walnut. And I'll spend a lot of time talking about the way they look. So I just thought I'd show you some that you didn't have to have pictures. A lot of oxalis are known for their succulent morphology. And so this is a species that has a succulent caudex, which is like the outer part of the stem. And then it also has these fabulous petioles and all kinds of cool. These are like bonsai oxalis. Some of them I've had in, well, Eve brought back from the Andes a long time ago, and we've had in the greenhouse, and I've had some impact on how they're shaped now. Most oxalis have 3 leaflets, so they have this morphology. But there's a group of oxalis that I'll talk about at the beginning of the talk mostly that also have 3 leaflets, but they don't have that trifoliolate morphology where they are all branching off at once. There's a stem between the first 2 leaflets and the terminal one. So that's kind of an interesting difference in oxalis morphology. Let's see here. Eve studies these ones from the Andes that have really succulent stems and kind of scrounge around the place. And some of them have developed succulent stems that they put underground, which we call tubers. And those have been domesticated in the Andes. There is a group of oxalis that occur in temperate regions around the world. So I'll explain that most oxalis are in South America, but some are kind of broadly distributed both in the Old and New World. And this is one example of that lineage. This is Oxalis oregana from Oregon. And it doesn't look right now very good. It's not very happy since I'm trying to get it to grow this time of year, I think. The oxalis that I have been most focused on, and the ones that are closest to my heart, are the bulb-bearing oxalis from the New World. So I've got a bunch here. Here's Oxalis macrocarpa, which I'll show a lot of later. It's really hairy. This is a really sad-looking Oxalis violacea, which I collected just a couple, like 45 minutes west of here. Looks a lot different in the field in Wisconsin if you've ever seen it. Some of the bulb-bearing oxalis have also evolved more than 3 leaflets, so they have these cool, like, pinwheel-looking leaves. All right, I think we'll get started. I think Eve is introducing.
Yeah. Good. Okay. We have the whole committee. So, so I will get started with the introduction. This is, well, obviously not as big a day for me as it is for Andy, but it is quite a bit of a milestone for me and for my lab, as well as for Andy, because this is the first finishing PhD student in my lab. This is a picture that we took right as soon as our lab was renovated. I wonder if it would show up better if I put the front lights down. That doesn't make any difference. Well, we can turn it down and that light is enough. So you can see it a little bit better. Here's a bunch of newbies in a brand newly renovated lab with a new professor and 3 new graduate students. And well, Katie decided to finish with a master's, but Jane and Andy have been with me from the very beginning when we first started in the fall of 2006. So we have learned an enormous amount together, and so this is really a milestone for the whole lab that Andy is defending today. So he's going to talk about his research. I'm not going to really talk very much about his research because he will do that himself. I'm just using this as an excuse to put in this cute picture of someplace in Brazil that Andy was doing field work with Magdalena Baio, who some of you may remember from last year, her visit to our lab, and Pedro Fiaschi from Brazil in one of his fieldwork expeditions. He had another one to Mexico. His first paper was published in American Journal of Botany a couple of years ago, and he will be talking about the other papers today. But I really want to be focusing on the other things that Andy's been doing, which include teaching innovation and mentoring and outreach. So one aspect of the teaching innovation was that way back in our second semester, Andy and Jane and I all took the Delta program's IMD, or Instruction Materials Development course, where we learned a lot about teaching, but we chose as our problem to work on the fact that it can be pretty difficult to explain plant life cycles to introductory botany students, especially because they're thinking very much of animal life cycles, which are pretty darn different. So we worked on some diagrams and some pedagogy. Jane worked more on the pedagogy, and Andy especially worked on developing the diagrams, and with the help of an undergraduate student who had a lot of HTML knowledge, developed the Life Cycle Interactive Explorer, which you can go see online. We feature a few different chosen plant life cycles, and there are pictures, and then there are word descriptions, and then there are ways to picture. Click on each of them to get to the other in sections, and then there's self-quizzes that the students can give themselves to make sure that they understand what's an archegonium and where does meiosis take place in this life cycle and things like that. So we presented this material back, all three of us, in the 2007 botany meetings in Chicago, and I think both Andy and Jane have been continuing to do assessments of these in different courses, both Botany 130, Botany 100. I think it was used at Edgewood for a while. So that has been, learning how to improve teaching has been a big aspect of Andy's work. He also learned in that course about doing things like the SALG, the Student Assessment of Learning Gains, which he's used in a number of different venues, including when he was the lecturer for anatomy. So he's been out front among us doing a lot of those assessment things. He was also the one in our lab, and I think the first lab in the department to develop a lab wiki. Now some other groups have lab wikis as well, but that was a good way to help communication from within the lab. So teaching innovations, also mentoring has been a really big aspect. Oh, this changed on the other computer. That's the thing, I wrote it on a PC and now some of the words disappeared. Okay, well, anyway, mentoring has been a huge focus of Andy's time here. He has taught an army of undergrads about laboratory techniques. This picture was at least 3 or 4 years old, so this is only the first 10 undergraduates that Andy taught in lab techniques, and I think the first 5 he taught as a group in a really efficient and innovative way to have a group of students that he took through the entire process of, here's how we extract DNA, here's how we do PCR, sequencing, aligning, and phylogenetic analysis, took them all the way through, and then there's been a lot of other undergrads since then. And the words that seem to have disappeared were about the mentoring group that Andy organized a few years ago with both graduate students and undergrads to help do peer mentoring among different students. I think it started as a way to have people teach other lab techniques that different labs might not know, but then evolved more into peer critiquing of abstracts and proposals and posters and talks and things like that. So that peer mentoring has been a really big focus of Andy's. Outreach, and some of you may know Andy mostly from the Evolution group. So he was a main component of the organizing committee for the Evolution Seminar Series for a while, and then has also been really active in Darwin Day events, so including being a regular Darwin Day volunteer, one of the helping to organize it, and then designing some of the best posters that Darwin Day has had over the years. I think this has got to be one of the most eye-catching of the posters. So he's been really active in that. So a few years ago, we nominated Andy for one of the university-wide awards, the Exceptional Service Award for graduate students. And we were really proud that he was the winner, and just looking at all the different things that I just described, you can see that he was really deserving of this Exceptional Service Award because of all this different mentoring and teaching, different activities that he's done over the years. So that's kind of all I was going to talk about. We'll let him talk about his own work soon. I just want to remind people that after he goes in there and the committee has at him for a while, we'll have a reception downstairs in 245, otherwise known as the back room. In a couple of hours after he gets done talking, the committee will have at him for a while, and then we'll have a reception down there. So you might, I don't know, maybe 4-ish you can start gathering down there, and we'll see when he gets let out. So anyway, Andy Gardner, it's really a proud moment for all of us.
Thank you, Eve, so much. So this is my title slide, and obviously, and I guess I'm gonna keep going back to 3 kind of main aspects of oxalis. Its morphology, the environments that it occupies, and also the geographies that it lives in. So I'm going to start the talk, though, with my acknowledgements, because if I try to finish it, I'll be in a puddle. So I'm really lucky, I think, to have received, well, I hope I will receive 2 degrees from this department. As an undergraduate here, I ran into like a lot of really awesome teachers and mentors, and this is already going downhill. So there are a lot of people that didn't like interact with me that much as a graduate student that I think really directly contributed to what I've done here, and I just wanted to thank them. The people in our lab are super awesome. We're a really diverse lab of people that have lots of different kinds of questions, and sometimes that's really hard because it means that it's hard for us to understand each other's research, but I think that's really great because it forces us to be more diverse scientists. We're sort of open to anything in our lab, and I think that's made us stronger. As Eve mentioned, a lot of the work I've done here has been with undergraduate researchers. So I was interested in mentorship from the beginning in part because of the mentors that I had here as an undergraduate. And I had the opportunity to oversee like the experiences of lots of undergrads over the years and to participate in their senior theses, which was pretty awesome. Some of them I spent more time with, some of them I spent less. I hope that they all got as much out of the experience as I did from that. Okay, so in the botany department, there have been a lot of staff and friends that have helped me along the way. They're always there for an answer to a question or some help when I need it, and that's really awesome because I know that everyone's behind me. Also, I want to thank the faculty on my committee and sort of almost on my committee. Dave, Ken, Tom, Brett, Eve. I'm really excited to talk to you guys after this and to learn what you guys think about it. You've offered me lots of help along the way, and I really appreciate that. Also, Ken and Cecile, who aren't on the committee formally, have offered just as much help along the way, especially with the comparative analyses. Marissa Otegi gave me the opportunity to be a lecturer as a botanist, and that was really awesome. And again, Eve, thanks. Okay, so the project that we embarked on was a really big one. We wanted to try to build a phylogeny for a huge genus that was spread across the whole world. And to do that, we had to engage with like a collaborative group. And we found a bunch of people around the world that were also interested in Oxalis. And that collaboration has really grown into a bunch of friendships. People like Stephen Weller have helped us by just being encouraging and offering plants and tissue. Others like Aaron and Arturo have helped by being guides in other countries, in their case Mexico. Pedro and Magdalena in that picture have been collaborators helping with tissue and also contributing to the sequencing efforts. And they also, we toured around southern Brazil together and collected, which is pretty awesome. Christoph Heibel and Kenneth Oberlander I've never met in person, but we've communicated over email, through the years so much, and they've both been extremely supportive and really helpful along the way. This kind of a systematic project requires the help of lots of staff at places like herbaria, where I need to go to see what's been collected of these plants before. So I wanted to thank those herbaria in the US and Mexico and in Brazil. Funding for the project came in large part because of the NSF grant that you've got, but also it's come from internal funds, departmental funds, and also some funds through some societies. Those are the things that made it possible for me to go to countries and collect these plants, and go to meetings and show off my research. The people that have walked with me down this path, thank you. There are a lot of them. Okay, and then my family, obviously. My grandparents and my aunt and uncle have been really encouraging. Also, Sasha's parents and brother, they've had to sort of watch me fool around on the computer on these weekends, and no idea what the heck I'm doing. My family, Evie, my sister, and Ray and Connie, who came today, they always want to know what I'm up to. And then, Sasha, she's come along for the ride. I don't think she anticipated that it would be like this necessarily, but I think we've both grown a lot together. I think most of the time her more than me. She always lets us take a whole day out of trips to go to a botanic garden. All right. Back to something that's actually easier to talk about. Okay, so I'm going to structure the talk into 3 parts. And they're all going to sort of revolve around this big question of how has this genus of plants persisted and diversified on the planet during its perhaps 16 million year existence. I'm going to start with a little prelude about how the world has changed and how ecosystems have become to be dominated by angiosperm flowering plants. And I'm going to introduce 2 big synthetic concepts. One, phylogenetic niche conservatism, the other, adaptive radiation. These 2 concepts kind of bring together lots of evolutionary processes, and they kind of sit on 2 ends of sort of a hypothetical spectrum of how plants can continue to diversify and do this big question. Okay, so after that little introduction, I'm going to talk about the whole genus, and I'm going to talk about data that's been collected both in our lab and also in the labs of our collaborators. And it's been my responsibility to assemble all those data and put out the first phylogeny for the genus, so that's that paper. And then after that, I'm going to focus in on the American bulb-bearing Oxalis, which are the clade closest to my heart. And I'll sort of use this to look at mechanisms for diversification that I saw evidence for in this one. Okay, so to the introduction. So if we're going to talk about the history of the world in relation to flowering plants, a reasonable time to start is about 150 million years ago. The reason for that is because that's probably about the time when angiosperms arose. There's a huge sort of confidence interval around that time period, and we're not sure if it was, perhaps earlier in the late Jurassic or maybe a little more recently. I know this because of studies that have increasingly incorporated more angiosperm diversity and more angiosperm fossils that allow people to calibrate the timing of diversification to history. So at this time, what we see in the world is that the massive supercontinents were starting to break up, and you can see a lot of green here. The world was much warmer, the world was generally a warmer and more forested place. So skip ahead to about 100 million years ago, and there's evidence that many of the clades of trees that dominate modern forests had begun to diversify. So we know that by about 100 million years ago, a lot of these forests were dominated by angiosperms. As the continents continue to drift apart and start to look more like we see them today, about 65 million years ago, we have the that event that happened off of what is now the Yucatan Peninsula that may have wiped out terrestrial dinosaurs. And also, this is probably about the time when oxalis lineages first started to arise. So I'll explain why we think that this is about when oxalis originated in a bit. Okay, so after the dinosaurs went extinct through now, there's been a generalized trend of cooling and aridification and increased seasonality in many parts of the world. So this has been driven in part by the fact that changes in positions of the climates have modified how ocean currents can drive both precip and temperature around the world, and also because of mountain orogeny. So many of the mountains, especially in the New World, are fairly young systems, especially the Andes in South America. And when mountains rise, they interrupt rainfall patterns and create wet zones and dry zones on either sides of them. So the world is basically getting a little more complex vertically, and with that we're seeing increased areas of seasonal rainfall and just dry areas in general. So with that, and especially as time goes on, we see that during the Miocene and subsequently a lot of lineages that appear to have what might be adaptations to these newer, more modern biomes start to really diversify. So here's a paper that's showing plants that we assume to be in sort of drier biomes, plants that are succulent in some cases, that are diversifying more recently. Some of these lineages have diversified by basically jumping to these biomes that they're prepared for, that maybe they had sort of some pre-adaptation to. So as these biomes have expanded, as there have become more grasslands, like temperate grasslands in the world, maybe that's opened up more opportunity for C4 grasses. On the other hand, other lineages may be shifting in a way where rather than just waiting for their biomes to expand and then jumping into those new areas, what they're doing is they're just shifting into adjacent biomes. And they're doing this by altering their morphologies or their physiologies through evolutionary processes And we assume that these could be adaptations to these new biomes. So this sort of sets up this question. On one side, many people are finding that evidence for lineages that are niche conservative. That is, maybe there are forest tree species that have been, that there's evidence that even long into time, those lineages were forest trees. And so maybe those lineages are niche conservative that can move more easily than they can evolve. And oftentimes we see these lineages on several continents with wide distributions. And studies have found that this might be sort of prevalent among angiosperms. On the other hand, there are lots of studies that highlight the role of adaptive radiation, which is kind of on the other end of the spectrum conceptually. So adaptive radiations are when a lineage is invading different biomes and evolving new morphology, perhaps to accommodate that. So in adaptively radiating lineages, we see frequent biome transitions and morphological evolution, and oftentimes adaptive radiations are sort of most obvious when they're in isolated environments, like island archipelagos. So island archipelagos are kind of classic models for for adaptive radiation, because we see that lineages will invade the islands and then they'll fill all the different niches available, kind of. So here's a classic paper in that, I think. And but we also see evidence of adaptive radiation, in continental systems and among the succulents. So the kind of conceptual framework here is Oxalis a lineage that's more on the side of niche conservatism, where it's sort of conservative in its biomes and its morphologies, but is able to just diversify by jumping into new geographies? Or is it more conservative in its geographies and more labile in its biomes and morphologies? Okay, so that's the framework. I'm going to talk about that in the context of the whole genus, and then we'll dig a little deeper. So for Oxalis, I'm going to start by building a phylogeny for the whole genus, a representative phylogeny, and ask, what are the major clades that we're seeing? How are these things organized on that tree? Then I'm going to ask questions about morphological evolution, biome evolution, and geographic evolution, and try to synthesize across those. So you can ask things like, how much of this morphology was innovated in South America, or did some of it innovate outside of the continent? Also, we can ask if this morphology arose like in synchrony with the arisal or the formation of these more modern biomes, or was it there first, or did it arise much later? Then I can compare the sort of the models of change in these characters that I'm talking about, and look at which of these characters are changing more frequently. Do we see evidence of more rapid shifts in biomes and morphologies in Oxalis than we do in geography, which would indicate adaptive radiation, or the inverse? And that's basically what I'm asking. Okay, so Oxalis is a really nice system for studying these kinds of big questions because it's big and diverse. There's a huge range of morphologies in Oxalis, from plants that are forming root tubers, true tubers, imbricate bulbs, what we call tunicate bulbs. There are creeping species. There are woody shrubs. There's a whole range of morphologies. And in addition, they occupy a wide range of geographic zones. So most Oxalis are in South America, most of the lineages. And on the map here, where I have the colors more intense, that means that there are more lineages and species in that region. So there are lots of species of Oxalis and lineages in eastern South America. In addition, there are lots along the Andes and in central Mexico. The other major diversification of oxalis is in southern Africa, where there are about 230 species of bulb-bearing oxalis. And then there are some oxalis that are indicated that are in temperate zones, both in the north and in the south, and some species that have become much more widely distributed, basically cosmopolitan. Okay, so these are 3 trees of basically all the collaborations data of oxalis that show the evolutionary histories we're inferring. These are parsimony strict consensus topologies, and so the branch lengths aren't so relevant. What I'm going to do is kind of walk through the tree, but first I'll explain them. This is a tree made of data from the chloroplasts, or the plastids of the plants. And this is a tree made from the nuclear data. And we keep them separate because there are times when they evolve, they have separate histories. There they are separately, and here they are combined. The main topological challenge that we have understanding oxalis evolution is that there's some disagreement among different loci and different analysis methods about the topology of early divergent oxalis. So either there are these clades that are together forming a clade, or they're separated into a grade at the base of the genus. Sometimes this one subgenus, which I'll talk about in a second, Thamnoxes, forms a clade, sometimes they're grade. So we can't talk with much confidence about the earliest divergences in oxalis evolution. So this first group of 3 clades down at the bottom there are all members of subgenus Thamnoxes. Oxalis is divided into 2 main subgenera, Thamnoxes and Oxalis. Thamnoxes is characterized by having these sort of woody stems. I have one here. It's a really little one, but they have woody stems, and they have these leaves that are mostly sort of pinnately compound, although there are several Thamnoxes lineages that have simple leaves, apparently. Thamnoxes are mostly in forests in eastern South America. Here's an Araucaria stand grading into a temperate grassland in Santa Catarina. That's Magdalena down there taking the picture. The next group that I'm going to talk about, which may or may not form a clade with the members of Thamnoxes, belong to the subgenus Oxalis. And actually everything else on this tree is in subgenus Oxalis. So these are the creeping Oxalis, and I didn't bring one in. Apparently I don't like them. So this is the clade of Oxalis that Magdalena has been most focused on. And this includes 2 members of 2 sections, Ripari and Corniculati. And they're mostly in eastern South America again. They all have this trifoliate leaf morphology, but some of them have, perhaps through shifts in their breeding systems, been able to kind of get all over the world. And I found this on Wikipedia, but I could have taken that picture like anywhere in the world. Okay, as we walk up into subgenus Oxalis, there are a clade or 2, sometimes forming a grade, sometimes clustering into clade, of species that are in northern and southern temperate areas. Subsection Oxalis, and here's Oxalis oregana. Come up and check it out later. They're not really succulent, but they do form these rhizomes and cruise along in the forest understory. They often dominate forest understories where they are. So here's oregana in California. You can see how different it looks than there. Acetosella / Montana, which we have in northern Wisconsin, looks basically the same. Moving up, there are basically 2 main big clades within the genus that I'm going to highlight. this first one is a group of 3 clades, which I call the Andean succulent lineages. These live in the Andes, and they have lots of different kinds of succulents. So most of the plants here that aren't bulbs are Andean succulent species, and among these are Oxalis tuberosa, Boca, but there are all kinds of crazy cushion plants and things like that. Up on top, this rest of the group, most of them form bulbs. So we tend to call that the bulb clade. But the bulb clade is basically made up of the New World bulb-bearing Oxalis, which I'll talk about a lot, and the African bulb-bearing Oxalis, which are up there on top. So that's kind of a broad look at the tree. Over here, the bulb-bearing Oxalis in the New World. And in Africa. So the next step after we build a tree to put that tree into some kind of geologic context, we need to use some kind of dating tool. And the way I'm dating Oxalis' history is by relying on the work of my collaborators. So Kenneth Oberlander, who works on African Oxalis, was interested in when those radiated. So he used several fossils from the whole family, the order Oxalidales, to infer that the African Oxalis are about 20 million years old, and that the whole genus is about 60 million years old. He has really wide confidence intervals on his dates because he was really cautious about sort of being cautious. Our other collaborators, Christoph Heibel and Suzanne Renner, just published their paper on Chilean oxalis. And they actually use kind of similar methods to calibrate their trees. And they found a fairly similar age for the genus, but with much smaller confidence intervals. So what I've done is I basically took the average and chose a confidence interval that encompassed all of those. And I set that as the age of oxalis. So when I do that, I build chronograms. And chronograms are phylogenies that are scaled to time. And so a chronogram gives us some idea of when these lineages are diversifying. So the thing that I'm showing here is this is about 60 million years. We see that some lineages of oxalis are doing major cladogenesis, in the Eocene and earlier in time, but that several lineages are doing cladogenesis much more recently in the Miocene and Pliocene. Right, so you can see that lineages have different histories of diversification timing. So the trees that I used to develop this, I used to do the ancestral state reconstruction, which I'll introduce now. So the first ancestral states that I've reconstructed are the geographic occupation of the ancestors of modern oxalis. So what I'm doing is I know the occupation of the current species, and then we can build a model and reconstruct the history of this trait. So what we see is that at the earliest nodes in oxalis, that we see the highest probability for an Eastern South American distribution. We also see some probability for a Southern Andean distribution. There are clades that are mostly Eastern South American, but then there's this big clade which may have originated in the Southern Andes early in its history. The African oxalis up top there, all got to Africa, they're the result of a monophyletic dispersal to Africa, probably rafting across the Atlantic over a much shorter distance than we have now. So when we model these geographies, we model an average transition rate that's quite low. I'll come back to those transition rates more and more. So turning now to morphology, I characterize oxalis morphology by succulents. So there are all kinds of different succulent morphologies. Some have succulent leaves, some have succulent stems, some have succulent roots, some aren't. Some have combinations of succulents, like root and leaf succulents. These are many of the American bulb-bearing species. And then there are some species that are just completely succulent everywhere. So when we model the evolution of succulents, we infer that early oxalis probably was not succulent, and that there are several clades down here that probably were also not ancestrally succulent. However, within these clades, we do see some shifts to succulent roots, and we infer those to be fairly recent transitions in the scope of things. On the other hand, this big clade here, we're modeling as being definitely succulent in its deepest ancestry. And so that implies that this big clade of the genus was ancestrally succulent, and the cool thing is it's been able to modify that succulent morphology a lot. So there are some clades of oxalis where you see lots of different colors on the tips. What that's called is homoplasy, and that's the result of lots of transitions in morphology over time. Some clades, however, within this, especially the bulb clades, look about the same color. And what you're seeing here is plants that either have succulent roots or not, but make bulbs. Maybe there's some inability for these to escape this morphology, whereas for these, there can be more labile. So average transition rates for succulents are way higher than geography. And when we model the transition rates between different succulent morphologies, stuff that's happening in here, we see those are even higher rates. The lowest rates that we're seeing are out of those bulb morphologies, basically. Okay, so on to biomes. Again, we have only a little bit of confidence about the earliest oxalis biomes. However, we have a lot of reason to believe it's probably mesic forest. That one has the highest modeled probability here, and also we know that many of these other biomes didn't exist, certainly not in much area in South America at the time that oxalis arose. So that also reduces the probability of these, and that's not something I could incorporate into this way of modeling these histories. So oxalis probably arose in forests, and several lineages of oxalis have stayed in forests. But oxalis has also gone back to forests on multiple occasions. And then you see all these other colors, which are these modern biomes, like arid areas, temperate grassland, and alpine zones. So here's Oxalis gigantea in the Atacama Desert of Chile. Here's Oxalis violacea up here in the temperate grassland, and Oxalis squamata at elevation in Chile. So we're also modeling higher rates of biome transitions than we are geographic transitions, but lower rates than morphology. The lowest, interestingly enough, the lowest average transition rates for biomes are leaving forests, and the highest rates are entering forests, which implies that maybe forests are both a source and a sink for oxalis lineages. Okay, so here are those three reconstructions of succulent morphology, biomes, and geography. We're modeling higher transition rates among morphology and biomes than we are geography. And that kind of serves to provide some evidence that maybe an adaptive radiation model is a better model for oxalis than a niche conservative model. And certainly for some of these clades, which have huge morphological and biome labiality, then we're definitely talking about an adaptive radiation scenario. One sort of piece that I can tack onto this is that we can look at diversification rates of plant lineages if we have a tree and we know about how many species fit onto the tips of that tree. So I've done this, and the main diversification rate that I'm modeling in oxalis, like rate change that I'm modeling in oxalis evolution, is here along this node. And so then what we can do is ask for, are there certain characteristics that are characteristic of that node and that group of plants? So here, succulent morphology kind of comes to mind right away. But also, these occupied the southern Andes. So it could have been either their geography or their morphology, perhaps, that may have led to their increased diversification. Most likely, it was probably a combination of the two. The second rate transition that we model is among the African oxalis. Not all of them, but just this core clade. And it's much harder to point to a clear promoter of diversification there. You see that they have really similar colors along there. I think that a lot of it has to do with their location. The Cape has promoted a lot of diversification in lots of lineages. And so it has to be something about that environment that differentiates it then. Okay, so I showed you that oxalis originated in eastern South America about 16 million years ago, and that while succulents has evolved many times in oxalis, one clade evolved it early and has manipulated it probably a lot more than other clades. The diversity of oxalis was generated in South America, and some of those lineages have dispersed in North America, Europe, and the rest of the world subsequently. Compared to biome and morphological shifts, we modeled much lower rates of geographic transitions, and this supports an adaptive nature, perhaps, to oxalis diversification rather than a niche conservatism argument. Okay, so we're through the prelude and the big introduction to oxalis. Now I'm going to use the American bulb-bearing oxalis to look at what are possible aspects of these plants that might be adaptive responses. And can we see a little bit more into the system of how these plants are adapting? So I'll start by talking about who the American bulb-bearing oxalis are and where their diversification has occurred. I'll talk a little bit about what we know about succulent morphological evolution. Then I'll switch to a discussion of basically how these morphologies and environments could be correlated with each other. So to start, I'm going to look at all of these morphologies and environments and see if there's evidence of any kind of phylogenetic signal in them, in their histories. And then I'll see if they're correlated with each other. Okay, so the American bulb-bearing oxalis and those trees you saw earlier are this group of plants, mostly bulbs, several different biomes, and quite diverse geographically for oxalis. Okay, so the first couple of slides are from the paper we published in January 2012. And this is pictures of plants that belong to this larger group. And most of them form bulbs or some kind of subterranean leaf-based derived storage structure. But some of them don't. So members of section Articulati form these like succulent stems, which we call codices. And when we wrote this paper, we thought that Oxalis linerantha didn't make anything underground. That was a storage structure. I went to Brazil and collected this plant with Magdalena and Pedro. And indeed, it does make bulbs. Bulbs here. And the more we collected, the more we found with lots of bulbs. And it turns out that the woman who described this plant, she looked at a lot of herbarium specimens, and I saw that she'd seen them, but she never saw an herbarium specimen that had bulbs on them. So, like, Just goes to show, check around underground before you put it in the bag. I've got that here. Okay, so what we found in that paper was that the American bulb-bearing oxalis are a big clade of bulb-bearing species that arose from an unclear relationship to these other things that kind of don't make bulbs. What we found is that the American bulb-bearing oxalis are in two main clades, one clade in Southeast and South America, and one clade in the Andes and into North America. We also found, interestingly enough, that this species, Oxalis triangularis, which is here, which forms these rhizomiform bulb structures, was nested among the rest of the species. So this plant and several other species were put into this section called pseudobulbosi, and earlier authors had presumed that maybe they were like early derived members of this group and that this represented sort of a transitional bulb morphology. So our results that put that among all these other bulb-bearing species suggested that that was not the case at all, but rather that this is a derived morphology from species that make more normal bulb morphologies. So that got me excited about morphology and climate in these plants. So what do we know about the relationships of succulent morphology and climate? We do know that succulent morphology could be a key innovation. There's the classic case of the ice plants in Africa, who've diversified exceptionally rapidly down there. Also, I just showed you that maybe succulence is synapomorphy for increased diversification in oxalis too. Also, there's lots of evidence from succulent lineages that succulent morphology has evolved several times in those lineages, and that it's also quite labile. So euphorbia, adenia, and oxalis all shift between different morphologies a lot. So is there any relationship between those morphologies and their environments? There are very few studies of this, but this one is most pertinent. This is geophytic plants in southern Africa, and these different charts represent different towns in southern Africa. So Luderlitz is near the Namib Desert in the driest parts of the country, and Grahamstown is to the east, also quite more dry, but Cape Town is where they get the most consistent winter rainfall and the most rainfall. So what they found is that plants that have geophytic morphology tend to have larger storage structures in these areas that get less rainfall and less consistent rainfall in the winter times. Presumably so that they can survive a year where they don't get enough rainfall, so they just don't do anything that year. So they found a little bit of a pattern, but it wasn't statistically significant, so they just calling it a trend. So the American bulb-bearing oxalis are a really good system, I think, for studying the evolution of geophytic morphology because they have such diversity. You've got all kinds of both above ground and below ground morphological diversity. Plants that make giant roots and big bulbs, plants that just have fibrous root systems like triangularis and these rhizomiform structures. So through the rest of the talk, basically, I'm going to talk about the evolution of these morphologies, and I'll highlight Oxalis macrocarpa and Oxalis triangularis because they kind of represent extremes of this morphological spectrum. You've got a rhizomiform species that's totally glabrous, it doesn't have any hairs on it, and it lives near rivers, and you've got a species that makes these big storage structures that lives in dry areas in western Mexico that's completely covered in chalk. So to start this comparative analysis, I built a new tree for the group. And this new tree just looks just like the old tree, except that it has a couple more things from that section pseudobulbosi inserted into it. And those came into this clade in a couple of different places. And so that is some additional evidence that these pseudobulbosy things are derived from normal bulb-bearing species. In addition, this is a dated phylogeny, which the other one wasn't. And it shows that this group is diversified in the late Miocene, and especially the North American species in the Pliocene. So it's a fairly recent expansion and diversification of this clade. So with that phylogeny, which will structure the comparative analyses, I have to add environmental data and morphological data. The environmental data come from herbarium specimens that either me or the authorities had checked their identifications and that we could georeference. So all the little dots on here are plants that I've seen, and that's where they came from. And what those dots are mapped onto are maps of this world climb data set, which basically has a bunch of layers that describe climates around the world. So you can see that the bulb-bearing oxalis in the Americas occupy areas from low to high elevation, areas that are generally pretty hot to areas that are generally much cooler over the year, and different precipitation environments, areas that get lots of annual rainfall to areas that get quite a little bit of annual rainfall, and areas that are highly seasonal, the red areas, where they only get a little bit of rainfall and it's only during a couple months of the year, areas that are fairly aseasonal where the rainfall is distributed throughout the year. So there's a wide range of environments, and to kind of tease apart how these environmental factors are impacting their niches, we use a principal components analysis. So a principal components analysis takes the data about environments and puts them together into these principal components, which are parts of the data that are correlated with each other. So here I'm plotting all the species by principal component scores 1 and 2. And principal component 1 is loaded with altitude and lots of temperature factors. So these plants are at lower elevation and higher temperatures, and these plants are at higher elevation and lower temperatures. Principal component 2 is loaded with a bunch of seasonality and precipitation factors. So up top we have more seasonal, and down here less seasonal. Here's less precip, there's more precip. One thing about these analyses is that a lot of environmental factors are loading onto each of the principal components, and that several principal components are necessary to explain the environmental variance that I'm characterizing. This is some indication that they're really parsing out their environments in a lot of different ways, and it's a really sort of multivariate environmental landscape that they occupy. Okay, so morphological data come from herbarium specimens. I can figure out what the diameter of a bulb is on there. But they also come from plants that I've brought back here and grown in a common garden. The reason we do that is because there's stuff like number of scales per millimeter, so that's how condensed the bulb is, that I can't get off an herbarium sheet. And also because if we grow plants in a common garden, that removes to some extent the effect of environmental plasticity effects on their morphology. So Oxalis macrocarpa here looks like this in March, but in July in Mexico, the plant is this tall. And it has a baseball-sized bulb. So again, I did a principal components analysis, and what we're seeing is that several morphological characteristics that are related to water storage and retention are correlated with each other, and that leaf and bulblet characteristics are on that axis. But in general, the morphological principal components analysis is kind of weakened because we have just a lot less data on morphology. Okay, so we can take these environmental data and the morphological data and look at how they've evolved each of these traits. And the way that I'm doing that here is to use something called Pagel's lambda. So what I'm showing is the original tree for these species, and then each of these columns is the score for a morphological factor. So most of them have 3 leaflets, some have like 12. And what you can see here is that there's a lot of diversity, and you can walk through and see where some things are quite different. You know, lots of trichomes on Macrocarpa and few on Triangularis. But when we look at the values for these, we can model their evolution on the tree. And by altering Pagel's lambda, we can get an idea of whether or not the values that we see are because of the underlying evolutionary history, or because maybe something else is influencing them. So most of the scores of Pagel's lambda that I model are quite low. So there's one here that has a Pagel's lambda score of 1, which implies that leaf area has evolved under basically a Brownian motion model of evolution, which indicates that there might actually be some of the evolutionary history that's influenced this character. Or at least that character is showing a mark of its evolutionary history, perhaps. On the other hand, most of the characters have lower scores of Pagel's lambda, and the reason, and what that implies is a tree where more of the branch length is associated with the tips than the internal nodes. And so what that's doing is, if this tree is a correlation matrix for these tip values, it's reducing the impact of the shared history and increasing the impact of the species time on their own branches. So most of the traits, we model a Pagel's lambda score of near 0, which implies a star phylogeny. That is, everyone basically arose at once, and they have no shared history. So what that indicates is that there's little evidence of niche conservatism, or there's little evidence of phylogenetic signal in these traits, and that maybe something else might be influencing them. The environmental data I also modeled quite low Pagel's lambda scores for. So I wondered, maybe they're related to each other. So you can look at environmental and morphological factors by just plotting them against each other. So here I've got like leaf dissection index, which is how dissected the leaf is, related to altitude, and you can see that it's just like a whole bunch of dots. There isn't necessarily a reason to believe that there would be a relationship between these. Some of them have kind of slightly more interesting relationships where there's maybe apparent thresholding, or cases where, you know, it seems that you can only have lots of leaflets in areas that are highly seasonal. What we're looking for, though, are relationships that look maybe more linear. And there aren't many. These are a little better, and they're relationships between storage morphology and precipitation factors. The ones in black are phylogenetically corrected regression models, and the ones in red are just uncorrected normal regressions. So we don't see any statistical support for these, but they do seem to indicate some relationships that might be relevant. And you can see how Macrocarpa and Triangularis are always on the kind of opposite ends of the spectrum of these. So just like Prochez et al., I don't really see a significant relationship between the morphology, like single morphological factors and environments. But I also saw that their environments and their morphologies are really multivariate. So what I'm doing next is to plot the principal component scores of morphology against environment. So these principal component values are those combined values. And we do see kind of a relationship between environmental principal component 2, which is seasonality and precip mostly, and morphological PC1, which is morphology related to storage structures. And so we see a statistically supported relationship there, and it has a very low R squared. But when we remove Oxalis violacea, which is kind of a latitudinal outlier, it gets a little better. Okay, so I've shown you that the American bulb-bearing oxalis are a clade of perennials that make bulb and rhizomiform structures, that they diversified in southeastern South America, the Andes, and North America. And then their niches and their morphologies are really complex, and also that there's little evidence of phylogenetic history that underlies that complexity. I saw that there might be some relationship between storage and water retention morphology and environmental factors related to water availability. And again, I found little evidence for niche conservatism in this clade, and I'm suggesting that this is probably an adaptively diversifying group of plants. So, there's my talk. I talked about the whole thing, and I tried to show you some patterns or some ways that lineages within might be accomplishing this adaptive radiation. So thank you so much for coming along on this ride with me, and I guess I really appreciate it. Thank you.
Will have plenty of chance to ask questions, but any of the rest of you who would like to ask questions, we have a couple minutes. If people have to leave, they can sneak out.
Yes, Susan.
Yeah, well, as an ecologist, I was interested to see this going into adaptive radiation and not so much phylogenetic constraints. And I was just wondering, this is in ecological time rather than evolutionary time, but do you have enough information on any one of your species to actually look at the environmental characteristics of individuals and sort of plot the variation of individuals or maybe clones or populations, however they're structured, versus and this would be after plasticity, but certainly the fact that your best effort, you got up to 25% of the variation. That's not bad in ecological terms, but where you might go after some of the current plasticity in individual species and see if that helps shed light on how you might structure some multivariate analysis to get a little more explanatory power.
So I think basically what you're bringing up is something that I think is one of the biggest weaknesses of that third paper. And it's that I'm not at all characterizing like within species diversity.
Yeah.
I'm just calculating averages and comparing those. And for some species I do have enough data that I could make an attempt at that. And I think to get it published, I will have to do that. But I'm not there yet. And for most of the species, I really don't have enough collection, and certainly not enough morphological data to try to see if there might be different ecomorphs or something like that. Okay.
So, how would you compare your results to this paper, where they showed so much phylogenetic conservatism? Is it how they sampled their species around the world?
I think that
Oxalis just kind of an outlier, or how would you recommend it?
It's my perception that the Chris paper sampled a lot of tree lineages, and I think that tree lineages are probably more likely to be operating in sort of a niche conservative framework. That being said, the methods that I use to assess that are really different from the Chris paper. Chris paper uses parsimony reconstructions, and they discuss transitions as unambiguous transitions between things under parsimony reconstruction, which tends to reduce the number of modeled transitions that you can have. Whereas I'm modeling rates and comparing those transition rates. So, I think that our methods are different, and to some extent that might be part of what's driving the different result. But I also think it is a relationship with the taxa that I'm working with.
A couple questions on the one of the last PCAs. You would have had such widely divergent units of measure, going from altitude to trichome length.
Yeah.
Is there a special kind of normalization of the?
So I'm normalizing all of the units to 1, to a range of 1.
Right, standard deviation of 1 and mean of 0, or?
Yeah.
Okay.
I think so.
Yeah.
And that has the effect
Then you just
of,
cross your fingers and run it and hope for a signal?
No, that's a standard way of doing it.
Yeah, I know, it just seems that these are really widely diverse units of measure. I don't know if there was a magical alternative to.
well, the thing is that, who knows what like a significant difference in morphology is. Like you can measure something that's a lot bigger, but that might not be nearly as important as like, some sheath, a bundle sheath
Yeah.
that allows it to do, C4 photosynthesis.
Sure.
It's hard to know what a useful unit is if you're trying to tease apart that variation. I love to, if you were comparing lots of different geophytes that had really similar morphologies, I think that there might be more opportunities to retain the original scaling, but
Yeah.
for this I chose to just scale everything to 1.
And my other question is, the genus, of course, is noted for oxalic acid.
Mm-hmm.
What might be the purpose of that, dominance throughout this genus? What is it doing? What function might it
Jane?
have? Is there a strong variation across the genus in the amount of it?
I think so.
I remember eating lots of this as a kid and enjoying the tartness of the.
It's enjoyed worldwide. Yeah. My perception is that there is a lot of diversity in oxalic acid production. Jane's work on Oka, is really focused on how oxalic acid levels and acid levels in general have been manipulated by human domestication, and she's found like really big differences, and they might be correlated with ploidy levels. Beyond that, I don't really know much at all.
I
about
set you up
oxalic.
well then.
Yeah, just wait some unspecified number of weeks and or months.
Yeah. Other question?
One last question. Have you tried to explore using, especially in the last part where you looked at these environmental and morphological features that clearly have a spectrum of values, but have you considered trying to put them into like 3 categories? One extreme, the other extreme, and then the middle part making them polymorphic, or and then running something like phase,
like a discrete analysis where
phase discrete.
To see if there's like the ones with the biggest bulbs that I put them into that category are often showing up in the areas that I've put into the category of most seasonal rainfall or least amount of annual.
Yeah, I'm using Bayes traits discrete, which will take into account phylogenetic.
So I did that actually a little bit a couple of years ago, just dumping things into these bins. And I did find a significant correlation using that tool. I really wanted to try to use as much quantitative data as I could in this, just because I was a little more interested in that aspect of the data.
Okay, well, I think the committee and Andy can all go across the hall in a few minutes. And the rest of you, I hope you will join us in a couple hours for a little reception down in 245.